screen. All right. Thank you. See if I can. Actually, maybe I'll put that like that. Okay. Hi, everybody. So we had stopped at kidney, or the kidneys, if I can get that going. Is that the way to do it, by the way, so it gets recorded? Right. Were there any questions from last time? It's kind of a few things. We talked about intestine and muscle. So the kidneys are kind of the last major organ that we'll talk about with amino acid metabolism. And you can change that to 2, because I'm really going to focus on the 2 most important, arginine and histidine. I'll show you in the next, in terms of the objectives here. And the rest, I think, are reasonable, so we'll keep those, let those stand. But the kidneys are important in amino acid metabolism, and they take up certain entities, amino acids up here and citrulline, so that just, I'll show you in a diagram in a second how those work. If you want to, you can bracket glutamine and alanine primarily being supplied by muscle, right, which we were just there. The muscle yields glutamine and alanine. And where's citrulline coming from? The small intestine, exactly. So citrulline's coming from the small intestine. The other thing I'll show you in our diagram is that the kidneys are a major site for synthesis of histidine and arginine. We already know, right? I could have thrown that out to remind you. From the citrulline, we're going to be making arginine. And then guanidoacetate, which you may or may not, has anyone ever heard of that? This is an important component of creatine synthesis. Where do you think about creatine? Anybody? Muscle, yeah. That high energy phosphate storage form that we keep in muscles so that when we are running to dodge the bullet or the bus or whatever, we've got creatine phosphate. So that's just another important thing that the kidneys do. So, the other thing about the kidneys that I would just maybe put here or add to this is the kidneys are the only other organ besides the liver where the gluconeogenic enzymes, the capacity to make glucose is, right? So the kidneys and the liver both have the full complement of enzymes needed to make glucose, gluconeogenesis. All right, I guess mostly I'm going to try to, I'll animate these and just show them to you, but I'm going to tell you don't really need to worry too much about the details on this because it's a little bit hard to follow. I had said that we make the kidneys preferential uptake of glycine and that they make, 3 amino acids are important. Glycine is taken up by the kidney to make serine, right? And these are non-essential to non-essential. When that happens, ammonia is fed into to be excreted. We do excrete some nitrogen as ammonia. I'm also showing that citrulline and aspartate get taken up. And if you think back to your urea cycle, which I hope you have refamiliarized yourself with, right? You maybe can realize that we need aspartate and citrulline in order to make arginine, right? Which I said is one of the major roles that the kidney has. We use some of the arginine and combine it with glycine to make that guanidoacetate, so that's part of how that works. This is showing arginine is one of the things that we ship out from the kidney, right? One of the major export products. And then this is showing how histidine gets basically produced from to, from carnosine. And then histidine is the other amino acid that we export. So I told you that histidine is conditionally essential. I mean, it is essential primarily. It's really essential for infants. We make a little bit of it, right? We still say it's essential for adults as well, but that's where the controversy comes, is that we can make it from carnosine. Okay, I said I wasn't going to go through that. I'm going to go ahead and animate all of this one. This is in a nutshell showing what I'm going to show in a larger diagram later, and that's how we make glucose, and we transaminate. We can take in glutamine and yield glutamate. And anyway, I'm going to leave that. Let me skip that, and then I'll get to it in a subsequent slide. It'll be easier to follow. Okay, you've seen this before. Citrulline coming into the kidney from the intestine, and all the two primary enzymes that are needed, and the aspartate that it takes up as well in order to do this. So this is just like what you learned with the urea cycle, except remember that we're really not making urea to any great extent. So that's the major point here, is that because ornithine has nowhere to go, you're not going to cleave very much of this arginine at all. So it allows for the possibility and the major pathway of releasing arginine for whole body use, because it's needed for protein synthesis elsewhere in the body. So, any questions about that? We had talked about that last time. Oh, I'm animated, sorry. Okay, whoops. So the other thing about just the kidneys and thinking about how our normal urine, what happens with nitrogenous wastes from protein metabolism. 80% of what we excrete in the urine of the nitrogen, from protein metabolism, is in the form of urea under normal conditions, normal physiologic conditions. 80% is urea. And again, sometimes people get confused, and I'll ask the question, do the kidneys make urea? The answer is no, okay? Remember that from that previous diagram. I don't want you to be confused by that, okay? So make sure liver is the only place that we make urea. That's where when we talk about urea synthesis, but we excrete at the kidneys, right? So 80% is as urea, very little in the form of amino acids, very little typically in the form of ammonia. However, when we get into a prolonged fast, under those conditions, then we start to excrete more as ammonia, okay? There's the other condition where we start to excrete more ammonia is what we call metabolic acidosis, when our blood pH drops below normal and we're considered in an acidic state. Okay, and so that's where we're going to go today. We're going to go there when we talk about interorgan glutamine metabolism, because it's related to ammonia excretion, if you can get there. I'll explain how. Okay, so normally 80% is urea. Under prolonged starvation conditions and metabolic acidosis, we start to excrete more like a 50-50 split between ammonia and urea. Okay? And I'll explain a little more as we go. So the kidneys are very important in a prolonged fast and starvation, and you can add metabolic acidosis as well. Be clear that those are two different physiologic conditions that just come together in certain ways in terms of how we excrete ammonia and some of the things with glutamine metabolism that we're going to talk about in a minute. So you can add and metabolic acidosis, but make sure that you, separate the concepts. So let's see. So one of the first, I think I've got it animated. Enzymes. So we've got glutaminase activity that is breaking down the glutamine that's coming in to glutamate. We release ammonia and we can excrete it like that, right? And this sequence, again, we start to shift to more like a 50-50 split, where a lot of the nitrogen is going out as ammonia, much more in that condition. So under these conditions, glutaminase activity is active, quite active, in order to take that glutamine and do that. And the other thing that's happening, the glutamate is also then going to be dehydrogenated to yield more ammonia and alpha-ketoglutarate. Alpha-ketoglutarate can be transaminated to yield pyruvate, which then is a major precursor, as you know, to make glucose. So it's kind of coming together where the kidneys are becoming a more important source under these prolonged fasting and into starvation kind of conditions, where the kidneys are making some glucose, and it becomes an important contributor of glucose to the body under these conditions. So 3 key enzymes involved. I got to click that too. Glutaminase, GDH. You can say ALT to make that simpler. There are other transaminases that may be feeding other amino nitrogen in here to get to ALT. Ultimately, ALT is what we need to get to pyruvate, right? But that's how we get amino acid nitrogen in there. And so the main points here under these conditions, we've got gluconeogenesis and the increased excretion of ammonia, which helps to correct the pH. And essentially, this is what I'm trying to show here, is that this is sort of an acid sink, or it's a way of taking protons and excreting them. Once we get them into the ammonium form, they go out. Ammonium so ammonia, when it's NH3, it's easily diffusible. Once it gets into ammonium ion, it's going to be then excreted. And that's a way for us to get rid of acid at the kidney under these conditions. So why, and actually I've got this on the next slide, I think. Do I? No, I don't. So when you're in a prolonged fast or starvation kind of condition, you are kind of acidotic because you're breaking down amino acids. And when you're in metabolic acidosis, separate from starvation, you're also, you've got to get rid of acid. So this is the way that the kidneys do that. They basically are compensating. It's not full compensation for, pH disorders, but it helps a lot. So are there any questions about that? Because that probably is something that you haven't heard before, I suspect, in biochem. So it helps to normalize the pH. There's another major reason. I just said amino acids breakdown, but in prolonged fasting, we don't do so much amino acid breakdown as we adapt, but there's another major source of acid that happens in a prolonged fast, starvation. Your body is producing. Can anyone think of that? What do we rely on, or what do we start making that helps meet the brain's need for energy under those conditions? Ketone bodies, right? So ketone bodies are another major reason why we get acid, a mild acidosis and starvation. Incidentally, I should just explain a little. Metabolic acidosis is something that doesn't, normal people walking around, normal meaning, healthy physiology, you're eating, that kind of thing, you're not going to be as acidotic from just eating something or changing what you eat. That's a condition that happens when you get out of balance because for whatever reason you're in the hospital, you've got an acute illness or something like that that changes your electrolyte. You can be acidotic mildly I shouldn't say that totally. So you can be ketotic, right? You can be producing ketones if you're following a particular type of diet strategy, right? What would that be? A low carb, really, really low carb diet, it's possible that you'd be in mild acidosis. But significant metabolic acidosis is really more like an acute illness condition or something that's driving, you're losing bicarbonate, you've got a pancreatic fistula or something like that. Okay. All right. So focus on histidine and arginine. Again, it is true, serine, histidine, and arginine, but I think it's the most important is histidine and arginine for being synthesized there. Are there any questions about these? Remember that this citrulline from the intestine being used by the kidneys to make arginine for whole body protein synthesis. That's sort of an example, I said, of inter-organ cooperativity, really. So the intestine and the kidneys are working together through complement enzymes, right? They don't have the full complement of urea synthetic enzymes, but by working together, they can provide arginine for whole body protein synthesis to meet our needs for it. And in a prolonged fast, gluconeogenesis becomes ever more important, and the kidneys help to correct the blood pH, and I'd ask you to know how. It's taking glutamine to excrete ammonia. And the nitrogen excretion under these conditions, fasting and metabolic acidosis, really becomes more like a 50-50 split between urea and ammonia. Okay, any questions about that? Because if not, we will move on to the last segment, which is some repetition, so bear with me, but the idea I want to talk about remind you, so we've talked about glutamine metabolism a lot in the liver, right? But there are other tissues that synthesize it. We've already talked about the muscle. There are tissues that synthesize it, and there are tissues that degrade it. The kidney, we've just talked about, is one that degrades it, and the intestine, right, we've talked about that uses it, glutamine. So I want you to think about those reactions. You know them already, so that'll be a review a little. And then how, under different physiologic conditions, our glutamine metabolism changes. And part of the story has already, we've started it by talking about the kidney. But it's an important thing to kind of, I think, keep in mind. You've seen this before, where you've got glutamine being broken down in the paraportal hepatocytes, glutamine synthesis in the paravenous hepatocytes. And I had, we've talked about how it's important that the liver has this backup protection, if you will, to catch whatever ammonia doesn't make it into being made into urea by the paraportal hepatocytes, and that ammonia that would be coming in from the intestine from metabolism of glutamine, right? We've got some ammonia coming in. It's important that it gets made to urea, but if it's not, if it escapes that, then we've got this backup system. So that's, in the liver, that's a really important thing. And incidentally, under those conditions where the kidneys are needing to get that glutamine to correct the pH, to get that ammonia to be excreted, the liver starts to ramp up its production of glutamine. So this enzyme activity actually increases under those conditions. So we're going to see a coordinate kind of increase of when glutaminase activity is high in the liver, glutamine synthetase activity tends to be low, and vice versa. So the liver can kind of control the supply of glutamine as needed to the kidneys. So glutamine synthetase, you know, I'm going to go through the different organs that have glutamine synthetase. And so, as we've just said, one of the purposes of glutamine synthetase, incidentally, can you remind me, did I remove these for you? Okay, thank you. So I'll let you jot that down. We've just said that, to make glutamine for export to other tissues, including the kidneys, right, are going to take it up. And it becomes important in an extended fast and in metabolic acidosis that this happens. And why do you think we have it in the muscle? What would be the purpose of having it in the muscle? We've talked a little about the role that the muscle has in making glutamine. So certain tissues that rely on it for energy might benefit from that production, from amino acid metabolism, right? Like the intestine and immune cells and that kind of thing, but the intestine's a big one. So for that, we've got major supplier of glutamine to the GI tract, small intestine particular, right? Small intestine, I should say. And it's also a way for the muscle to get rid of that ammonia that comes from amino acid metabolism. If it didn't have that way, that ability, we'd have toxic effects in the muscle, right? Because ammonia is toxic. So amino acid metabolism produces ammonia. We got to have a way of packaging it, right? What do you think the brain, what purpose would the brain have in having glutamine synthetase, would you guess? Say it, whoever's. Yeah, you want to protect the brain. Very good, thanks. Crystal, right? Is that? I don't have my glasses on. So, you know, to detoxify ammonia from amino acid metabolism and potentially production from other neurotransmitters. We've got monoamines and other things like that. A lot of activity in the brain, and as those get degraded, or you know, it's important that we have a way of detoxifying. All right. What's the purpose of glutaminase in the paraportal hepatocytes in the liver? What does glutaminase activity help us do at the liver in the paraportal hepatocytes? Sorry? Right, helps us get amino nitrogen into the urea cycle, right? So we can excrete it. Now, how about the kidney? What's the kidneys? What are the kidneys doing with glutaminase activity? Why don't you finish jotting? Anybody? Well, we talked about it particularly in a prolonged fast, right? You can look back in your notes. It's okay. Do that. What's it doing? What is glutaminase activity helping us do at the kidneys, guys? Yeah, Crystal? Exactly. It is helping us. It's basically breaking glutamine down, which is coming in from other tissues. Under these conditions, kind of, we're bringing in more glutamine so that ammonia gets released and we can excrete hydrogen ions as ammonium ion. We combine the ammonia with the hydrogen ion, right, to get rid of acid. So we're trying to correct the pH under these conditions to some extent, okay? So is that clear? Was that, I need to go over that again, how that works. So if you look back at that diagram that I gave you for the kidneys, a couple slides or a few slides back, glutamine is actually doing two things, really. It's helping, the carbon from the glutamine is feeding into gluconeogenesis in a way, right? Because there are, you're deaminating it. You yield glutamate, and then you go through transamination, and you're helping to get that carbon to the form of pyruvate. And it's releasing ammonia to be combined with acid to excrete acid, essentially, as ammonium. Small intestine, I'm just going to click that. We use, the small intestine relies on glutamine for energy, so it absolutely has glutaminase activity is going all the time in the intestine, okay? And then similarly, immune cells are to use it for energy, right? So they use a lot of glutamine for energy as well. Does any of that not make sense to you at this moment? Or you have a question about any of that? So some things, some activity, enzyme activity, is kind of constitutive, if you will. It's kind of going all the time. And the intestine is going to use glutamine wherever it gets it from, right? If you're in the fed state, a lot of it's coming from the diet. If you're in the fasted state, it's going to be coming from muscle. It's going to be coming from liver. Paravenous cells are going to be supplying the intestine, but it's going to be doing glutaminase all the time. Kidney, it's only under conditions where you need to be addressing that pH issue, and that you might need, you've got the need for gluconeogenesis to be contributing. It's sort of a combination thing, but a lot of it is driven by that acid, correcting the acid balance. And then paraportal and paravenous are going to coordinately regulate those activities depending on physiologic state. You've seen this reaction. Just remember it's ATP demanding. Key points about glutamine, to add to what we've already said. We've said it's the major carrier of nitrogen. The other thing is it readily diffuses across tissues into and out of the blood and into and out of cells quite easily. Oh, I know what I'm sorry to go back. Let me go back just a second. The other point I wanted to just remind you that we had said before about glutamine synthetase is the thing about its Km. Or its affinity for ammonia. What do you remember about that, glutamine synthetase? It's a high affinity, low Km? Yep, exactly. So it's got a, for ammonia, even if you have low concentrations, you'll have this enzyme active. High affinity, low Km enzyme. Okay, key points. Readily diffuses, major carrier of nitrogen. You already know this. Primary energy source for small intestine cells and immune cells, right? Macrophages and other things. We've talked about it. It plays an important role in acid-base regulation. And then glutamine synthesis in muscle. It's important in fed and fasted states. It's a major producer of glutamine, primarily from branched-chain amino acid metabolism that's going on in the muscle, right? The source of those amino acids to muscle in the fed state are what? Fed versus fasted. What's the source for branched-chain amino acids? In the fed state, it's going to be diet, right? Obviously. And in the fasted state, what is the source for branched-chain amino acid metabolism in muscle? Body protein, right? Muscle protein and degradation. Okay. This busy slide, it's a little bit busy, is showing where glutamine synthetase is working. So don't be worried. I mean, I'm just writing out the reactions. It's the same reactions. If you know the reaction, I could have done this a little simpler. But brain has got it, right? Glutamine synthetase is going on in the muscle from amino acid metabolism, and paravenous hepatocytes could be doing it as well. And again, when this is active, it's coordinately regulated with the other cells that break it down, right? So if this is active, glutaminase won't be. And then glutaminase activity, this is just showing breakdown of glutamine and for what purpose. It's different in each tissue. We talked about the liver for urea synthesis, for the kidneys under those conditions that we talked about, prolonged fast and acidosis, you're excreting ammonia, which is basically acid, which I'm not showing the full reaction. And then intestine and immune cells are always using, they're using glutaminase to help them use glutamine for energy. Okay, so those are just some basic. And here I'm just showing the intestine produces alanine, and I've got various things animated here. We talked about what else is happening in the kidney under those conditions. We're making glucose, gluconeogenesis is going on, and the intestine releases alanine that can be used also by the liver cells for making glucose or other things when needed. Okay, what else do I have here? I've got a few things that are probably a little, yeah, so, yeah, I always think of trying to make this easy for you to study, so I apologize because it is a lot repetitive. I feel like I'm always repeating myself, so that is boring. So what I've tried to do then is just to kind of put it together a little by tissue, right? Just to kind of remind you again what each tissue is doing with glutamine. So I'll click through this. We've talked about these points up here. And then we've also talked about this. How would you answer this? Which physiologic conditions does the intestine, do you think, increase its uptake of glutamine from the blood? And so thinking about the fact that the intestine relies on it for energy, so what's it doing? When would it have to take more proportionally from blood as, to meet its needs? Fasting, right? Totally. Under those conditions of extended fast, and incidentally, under acidotic conditions, or not acidotic, metabolic stress is another condition. And so this is where clinical meets metabolism, right? In somebody who undergoes a major injury, they fall off the roof or whatever, they get hit by a car, they get injured, they're going to be in the ICU, they're going to have a metabolic stress reaction. And under those conditions, your intestine, it's like a shock reaction. We talk about it in MNT, how it's so, we've learned that it's really important to feed your intestine as early as possible after injury, right? Because if you don't, the intestine, it is an immune function, an immune organ, really. It's got a lot of barrier protections, and so if we let it break down, we're at risk for a lot of complications. So the intestine, in some ways, it's probably protective for it. It starts drawing more glutamine from the blood. We may or may not be able to actually meet that need by the liver under those conditions, but anyway, so different metabolic conditions, but similar in terms of the intestine drawing more glutamine. Okay, the liver glutaminase, it's active in the fed state, right? Remember that? I told you that urea cycle and glutaminase activities are active at the same time. And incidentally, this is a new piece of information amidst all the repetition. Metabolic alkalosis, which is the opposite of acidosis, you've got glutaminase activity also active under those conditions. So even though they're separate physiologically, very separate in this case, right? Fasting and acidosis, we've got a little bit of overlap, can be. We can also have acidosis completely independent of, you can be fed and have acidosis, right? In this case, in the fed state, glutaminase is active, and also in metabolic alkalosis, okay? And it's all, that piece of it's about the pH. So the significance of this is really up here. We've already talked about the significance of glutaminase because it's feeding amino nitrogen into urea, right? Under this condition, we now don't want to be excreting acid. We want to be not doing that at the kidney, right? So it's just the opposite kind of the significance of it, of the opposite activity of the kidney under those acidotic conditions. In alkalosis, we want to hold on to and excrete more in the form of urea. So we're not getting rid of acid. That's the significance of that. Under these conditions, when glutaminase is active in the liver, kidney glutaminase is shut down, if you will, or not active. Not active is probably the way to think about it. So the kidney glutaminase and the liver glutaminase are coordinately, in opposite ways, regulated under these conditions. So essentially you could say, and some people have, that the kidney and the liver are kind of inter-organ cooperativity, another example of inter-organ cooperativity, because the kidney glutaminase and the liver glutaminase activities are not active at the same time. Liver is active, kidney is not. So that it allows for that correction of pH. Let that sink in for a second. Are there any questions about that? you want to ask, you guys? Is it okay? I mean, I don't mean to, can I go over it again? So glutaminase activity, remember what it's doing in the liver, right? Is it's releasing ammonia that goes right into making urea. So we're going to be making more, proportionally, we're going to be excreting more nitrogen as urea. Remember, under these conditions, when you're in fed, let's talk about it because we've really talked a lot about feeding and, you know, normal physiology, right? Don't think about the pH issue. In the fed state, you've got lots of amino acid nitrogen coming in, you're making urea. 80% of what we excrete as of the nitrogen from amino acid metabolism under those conditions gets excreted as urea. Well, in alkalosis, we excrete more as urea as well, less as, we don't want to be excreting ammonia at the kidney, which is, you know, kidney glutaminase, that's its whole purpose. Under conditions of acidosis, we want to get rid of acid at the kidney. When we're in alkalosis, we want to hold on to acid. Does that make sense? So this is without really talking all about acid-base balance, which is more complex than this, but the liver and the kidney are coordinating. They're working together basically to get more of the nitrogen out as urea. So at the liver, it's making lots of urea and not so much yielding. It's not that ammonia that gets released from this goes immediately into urea. It's not going anywhere else, if that makes sense. Better? Okay, I know it's confusing. Here I'm showing what's happening at the kidney. Glutaminase is activated, so, you know, glutaminase active in a fast, in an acidosis. Here the kind of, in a nutshell, let me click all of it. The reactions that are going on at the kidney, this maybe helps to gel that a little bit from what we just talked about. Only in acidosis and prolonged fasting is this really going, where we're making glucose, we're breaking glutamine down to get ammonia out as a sink for acid. We excrete it then as ammonium. And under these conditions, we up the nitrogen going out as ammonium ion, like 50-50 compared to urea. Any questions about that? On the other hand, now I've got three, I think I just do one slide on glutamine synthetase. You've seen when we need to be making glutamine to supply it to the kidney under those conditions where we want to get glutamine to the kidney so it can help us get rid of acid, then the liver paravenous cells ramp up their activity and make more glutamine and less urea by periportal hepatocytes. Okay. The significance of this is what I've said previously, which is that you're supplying glutamine to the kidneys so that you can correct that pH under these conditions. Is there any question about that, significance of it, how it works, whatever? Well, you're tired of hearing the word glutamine. Okay. We talked about this a moment ago. Why does metabolic acidosis occur with fasting? Ketone bodies in a big way. Initially, protein catabolism, we get pretty good at conserving protein over time into an extended fast, but there is some acid produced from that as well. And how does this, or I'm not going to go over it again. I want you to just be able to, in your mind, talk yourself through that. Okay, how does it work where you've got coordinate regulation of those enzymes to help us correct the pH under those conditions. And then I've just tried to encapsulate it for you, showing you what's going on in that at an organ level in terms of enzyme activities. Acidosis or extended fast, liver is cranking out glutamine, right? Muscle is also producing glutamine like it does. This is so confusing. Kidney glutaminase is active. That's the main thing. Okay, if that helps. I'm trying not to make it more confusing by explaining it, but hopefully this helps you think about that under an extended fast, what's happening. Any questions about this while does it make sense? A bit early. There's my summary slide. Again, trying to help you study for this. Hope it makes sense. And I'm not going to read it to you, but any questions about it? Does it make sense as you're kind of think that'll help you keep it clear? Okay, new information, or a little bit more about metabolic stress. Again, that response to major injury, not hopefully the stress. I hope that you're not feeling too much stress about the little quiz that's coming up, okay? Although the week I've been having, I do feel like it's metabolic stress. I don't know. So I know probably you feel that way. But metabolic stress that we're talking about here is your body's mounting that response, major response of kind of hunkering down and responding to traumatic injury, blood loss, trying to keep yourself from having infection, massive systemic infection, et cetera. Under those conditions, I said the intestine takes up more glutamine. And then I said that often we don't meet the need. So paravenous hepatocytes do what they can. They're trying to make glutamine, and it's not enough. So what we tend to see is this drop in plasma and tissue concentrations of glutamine. So physiologically, that happens. You've got muscle catabolism going on at the same time. We get changes in the GI tract and the immune cells, so we've done some work. Some studies have come, and there is some evidence in some populations that glutamine supplementation can actually be helpful in the clinical setting. Those of you that are in MNT, we've got some guidelines about glutamine that I didn't point us to, that we feel free, those of you that have access, go check it out, because it's kind of a little disappointing, I'm going to say. So as we've had some randomized controlled trials, sometimes it's good and sometimes it doesn't really pan out, even though the theory is really solid. You think about the fact that the plasma levels drop, and you would think that giving it would be unilaterally helpful, but it's not always. So there's probably some physiologic reason why that happens that may be beneficial to the body in some conditions. So there's some very small evidence. Short bowel syndrome, it's a little bit spotty. Bone marrow transplant, very little studies, but some that have suggested it. And then traumatic brain injury, trauma, burn injury, a little bit more stronger evidence for that. And each of in MNT we'll talk a little bit more about that. And that is the end of the glutamine story, and I won't mention the word glutamine again for at least a little while. You've been very patient with me. I know it's a little bit, I always feel like I'm probably overly repetitive, so forgive me for that. Do you have any questions about anything? And I think you've got a quiz coming, so you've got plenty of time, right, for the quiz. That's good. I'm amazed, actually. Sometimes it really takes me a lot longer to get through this, so I didn't repeat myself maybe as much as I thought I had, but no questions for me? It's been fun. Thanks for, letting me come and talk to you about a favorite topic, amino acid metabolism, and see the rest of you in MNT. All right. Thank you. All right.